Annyeong! Hi, it's me, Juvil. Let's talk about biochemistry. So, what is biochemistry? Is it biology or a chemistry? Well, as might you guess, quite literary terms of sciences. In a great panoply of the sciences, biochemistry sits right here. If we go smaller, we get into a small molecules made of just atoms, which is chemistry. And even smaller than we get to the particles that are smaller than a single atoms, and that's a particle of physics. If we instead go much bigger, we are looking no longer at individual molecules, but entire cell or a part of a cell. That's biology. Right in between in this biochemistry, these large biomolecules like proteins and DNA and the things they can do. Now, what exactly is our point in our video presentation that we're going to say today? This refers to new, unique discoveries related to our marine fisheries or new research discovered by our great biologists or scientific studies and analyses that this year or in this year 2010 and the present. So, let's find out.
So the title of the research is Effects of Dietary Protein and Lipid Levels on Growth, Body Composition, Blood Biochemistry, Antioxidant Capacity, and Ammonia Excretion of European Grilling. So what is the information of the title? So this study evaluated growth, body composition, antioxidant capacity, innate immunity, and ammonia excretion of European grilling with diets containing different protein and lipid contents. So, who are the authors? So, the authors are Samad Rahaminejan, Keann Branzuela, Marisol Izquierdo, Alexander Molinovsky, Jitka Kolarova, and Thomas Polgar. So, it was published on 13th of July 2021. So they conduct the study in Faculty of Fisheries and Protection of Waters, University of South Bohemia, Bunani, Czech Republic. And the instruments they use are sampling and analytical and statistical analysis. And the DOI is presented here on the screen. The analysis and the strong and weak parts of the research and so the strong part is that these results suggested that 40% of protein can support growth of European grilling with an average body weight of 25 to 60 grams, and increasing dietary and fat content to 20% can enhance feeding efficiency and protein efficiency ratio.
Let's talk about the two published research, the international research and the local research. First, let's tackle the international research. The international research titled Influence of Nitrogen, Limitation and Lipid Accumulation in EPA and DHA content in 4 marine microalgae for possible use in aquafeeds. Information on what is the title. In the present study, the effect of nitrogen limitation on the lipid content and fatty acid profile in different lipid classes of adactylum trichornotome, Isochrysis AFF, galbanoclone 3, ISO, Rhodomonas baltica, and Nanochloropsis oceanica were investigated. The author of this research is Xinxing Wang, Healthy Caroline Fox, usually Matilda Scoggin Colton, Olaf Vanstein, Kjell Ingritan. This research is published on March 12, 2019. Conduct the study in Department of Biology, Norwegian University of Science and Technology, or NTNU, Trondheim, Norway 2, Department of Biotechnology and Food Science, Norwegian University of Science and Technology, or NTNU, Trondheim, Norway 3, SINTEF Ocean, Trondheim, Norway. They use sample and data analysis as the instrument. Analysis on the strong and weak part of the research and why. In the strong part, P. trichornotum had the highest EPA content, while N-oceanica showed the highest EPA productivity due to the high content of lipid. The highest DHA productivity was found in Isochrysis AFF galvanoclone TISO from butyrate and limitation, mainly due to the high biomass productivity. In the weak part, as shown in the present study, increased lipid content in microalgal cells due to strong and limitation induction may not increase the lipid productivity because biomass production is usually reduced. Now let's proceed to the local research, titled DNA Barcodes of Caulerpa species from the Northern Philippines. Information on what is the title. The core subject of the study was to make use of DNA barcodes to assess the diversity of Caulerpa in the Northern Philippines. The present study aimed to identify Caulerpa collections from Northern Philippines aid with DNA barcodes. Tufa gene sequence data of recent intertidal and shallow subtidal collection confirmed the presence of 7 genetically recognized Caulerpa species in the Northern Philippines. The authors of this research is Richard B. Dumilag, Zayzay A. Aguinaldo, Ver Angelo M. Alcoriza, Maria Pia Sara B. Balucanag, Alessio Rose T. Dulalia, Arnel R. Sayasa. This local research is published on June 2, 2019. They conduct the study in Department of Biological Sciences, Marine Genomic and Molecular Genetic Laboratory, Marizon City, Philippines. They use intertidal and shallow subtidal collection, DNA extraction, and Tufa gene application, DNA barcode analysis as the instrument of this study. Analysis on the strong and weak part of the research and why. Strong part is they provide a map and image where the said species is taken, but the weak part is that they still need more information to confirm the other unconfirmed species in Northern Philippines.
So now let's proceed to the next research. As you can see in the screen, those are the topics that I'm going to discuss. But first, let's talk about the international research entitled Lipids in Prokaryotic Origin at the Base of Marine Food Webs. The base of the food web is composed of bacteria and archaea that survive and thrive under the most extreme conditions. The present work aims to provide a comprehensive review on lipid production in bacteria and archaea and to discuss how their lipids and both heterotrophic and chemoautotrophic origin contribute to marine food webs. This research will not be accomplished without this author, Carla R. D. Carvalho and Maria Jose Caramuj, and it was published on November 29, 2012. They conduct the study in Institute for Biotechnology and Bioengineering and Center for Environmental Biology in Lisbon, Portugal. The instrument used are the statistical data analysis. The UR number is seen in lower right of the screen. So the strong part of this research are the result are compromised to the expectation of the researchers, while the weak part is mainly abbreviation US use. The number, percentage, etc. of the research is not being put in a proper table where other people will understand easily. So now we'll move on local research entitled first deoxyribonucleic acid or the DNA evidence of the occurrence of Pacific bluefin tuna in northern Philippine waters. In this study, Pacific bluefin tuna caught north of Polillo Island in the Philippine Pacific seaboard and was identified through DNA barcoding using mitochondrial Delos region and cytochrome oxidase 1 gene. Analysis of the generated Delos sequence and partial sequence from the Philippine bluefin tuna using blast search showed 99% and 100% similarity, respectively, with published reference sequence for T. orientalis. Also, the studies will not be accomplished without these authors: Katrina Pierce Sarmiento, Minerva Fatime H. Ventolero, Rafael V. Remesca, William S. De La Cruz, Mujikiwis D. Santos, and also it was published on May 25, 2016. They conduct the studies in North Polillo Island in the Philippine Pacific seaboard. Instrument used is sampling method and DNA extraction. DOI number is also seen in lower right of the screen. The strong part of the research is they prove that there has a bluefin tuna occurring in a specific place with the result of the DNA, but the image of DNA result of the species is not shown. They only say the percentage of DNA extraction in the paper, which become the paper is half-truth.
International research. This research entitled Lipids in Marine Ecosystems. This research concerns compounds that can be operationally defined as lipids. The basis of this definition is their extractability in nonpolar organic solvent, which provides a convenient means of separating them from other compounds in an aqueous matrix. The author of this research is Christopher C. Parrish, and it was published in April 22, 2013. He conducted their study in Canada. The researcher used experimental instruments in their study. The DOI number can be seen in the screen. So let's proceed to the strong and weak parts of the research and why. So the strength of this research is data sets of lipids from marine ecosystems are often interpreted using chemometrics as they permit data reduction and objective interpretation of the results. Lipids provide the densest form of energy in marine ecosystems. The weak part of this research is the application of bulk and compound-specific stable isotope mass spectrometer methodology, and its modeling should be investigated further for both fatty acid and sterol work. So let's proceed to local research, and this research entitled the fractionation and quantitative analysis of different components of Suzuki-Trium sp or POL1 strain. This study was conducted to search for microorganisms rich in DHA or docosahexaenoic acid, such as stochochytrids as alternative commercial source. Fatty acids compositions and distribution of lipid components of Suzuki-Trium sp or POL1 were harvested. Was studied with special emphasis on the distribution of docosahexaenoic acid or DHA. The authors of this research are Modabel May in Apau and Jose M. Oclaret. It was published in October 28, 2012. They conduct their study in Polo Dapitan City of Western Mindanao. The instruments used are experimental and statistical analysis. The DOI number can be seen in the screen. The strong parts of the research are the Philippine stochochytrid was found to be better alternative sources of DHA than of fish. Also, the researcher is able to quantify and analyze the lipid fractions of Suzuki-Trium sp or POL1 or fatty acids components and to identify which lipid fraction has promising DHA levels. And the weak part of this research is that they cannot provide explanations or deep insights into why something is happening.
So let's have first international research by the title Detection of a Diverse Marine Fish Fauna Using Environmental DNA from Seawater Samples. So what is all about this research? In this study, it presents the first recording of marine fish biodiversity using eDNA from seawater samples. This study provides the first evidence that a very simple eDNA-based survey may offer a coverage of local marine fish faunas, which is comparatively better than or at least as good as any single conventional method. Together with these others, Philip Francis Thompson, Jos Kilgast, Lars Landsman Iverson, Peter Rast Moller, Morten Rasseman, and S.K. Wellerslev. It was published on 29th of August, year 2012. They conduct the research in University of Connecticut, United States of America. So as a student, the instrument that I use is to find this research is internet, while the real researcher of this are the sampling locality where they conduct and made the research, water sampling, DNA extraction, and PCR amplification. The strong part of this research is, although the further studies are needed to validate the eDNA approach in varying environmental conditions, our findings provide a strong proof of concept with a great perspective for future monitoring of marine biodiversity and resources. While the weak part of this marine ecosystem worldwide are under threat, with many fish species and populations suffering from human overexploitation And let's move for the local research with the title Two New Species of Freshwater Gabi Fish Discovered in Palawan, Philippines. So what is this research all about? Biologists have found two new species gabi fish in Palawan, a Philippine archipelago. The gabi fish is both belonged to the genus Rhinogobius. Together with the authors Ken Maeda, Chuya Shinzato, Ryo Kuyanagi, Taiga Kunishima, Hirozumi Kuyabayashi, Niryoki Sato, and Hermini P. Palya, together with the school at Okinawa Institute of Science and Technology, or OIST, a graduate of university. It was published on the 15th of November 2021. The research conducted at Palawan, Philippines. And again, the instrument that I use is internet to find this research. And the real researchers, they use our sampling locality and a personal survey, where they personally go to the place where the species discovered. And what is the strong part of this research is that a team of biologists from the Okinawa Institute of Science and Technology, or OIST, in Japan, and Western Philippine University, or WPU, in the Philippines have found two new species of gabi fish in Palawan, a Philippine archipelago. The gabi fish both belong to the genus Rhinogobius, were described recently in the Journal of Zooxtaza. And so far, there is no weak part of this research. Thank you.
So my future plans about fisheries and fish farming is to research and formulate diets that are highly digestible and produce less waste, thus improving food conversion ratio and fish farming. That's all.
The future study should be more focused on new discoveries of species in the Philippines. Such identification adds to the diversity of various species recorded in the country, but the plan is it should support the previous proposal of the Comprehensive National Fisheries Industry Development Plan that considers any other species as one of the species which could contribute to the sustainability of the commercial fisheries subsector in the Philippines. The confirmation of the existence of a newly species in the Philippines water should be more become the highlights now that will surely contribute to the important role of the Philippines in conserving and sustaining this commercially important but vulnerable species together with the other ranger states. They should prioritize more conserving this species in order for the fishermen in the Philippines have benefit in it.
What is my thought about this marine fisheries is that for me, if I were to be a person also with discoveries, strange in all the discoveries around the world, that would be tremendous and a very privilege to you, because you are the one recognized person who discovers these things. So it is tremendous reward for to yourself, because for the future generation, it will be available to the students, which will give them more knowledge. So, in my research that I read and know, I can say that all the old things are not really found that makes us give information and add history that to our world that we should study, and especially the strange or a new things that should be examined by experts. Especially in our ocean, there are more or many more animals or mysterious creatures that live in underwater that have not yet to be discovered. My interest here is how extensive marine fisheries are, where there is a lot of information. We should learn about it, also add knowledge to us, especially we marine fisheries. I really should know all the types, all the forms, all the new things we get in the marines.
To my future plan on research. As a student, I am interested in fish breeding, and I think improved breeds show increased potential for fishery aquaculture. In our culture, fish breeding is a very important activity to ensure the availability of fingerlings for the stocking of ponds, pens, tanks, and cages. The benefits of fish breeding may include improved growth, resistance to disease, or robustness in diverse farming environments. That is why my future plan is to study and make a research paper on how important fish breeding is when it comes to fish management and fish conservation in order to enhance fish health and sustainable production.
If I were conducting a research in the future, I plan to conduct a research about the daily activities of a certain species like seahorse because I want to discover those things on my own and say that it is true. Because I see it on my own eyes. I didn't say that the research of those researchers is not true, but I love to discover it on my own and say how amazing it is.
And that's all the 5 lists of international research and 5 local research for today's video. Thank you for watching and don't forget to like, comment, and hit the red bell button to subscribe our YouTube channel. Thank you for watching. Bye!